Welcome everyone to this week's data science seminar. I'm the host, David Bader. I'm a distinguished professor at the New Jersey Institute of Technology, where I direct the Institute for Data Science. This is our last seminar of the academic year, and we appreciate everyone who has watched many of these seminars all year long. And we saved the best for last. I'm really pleased today with our speaker, Dr. Joseph JaJa. And during the seminar, if you have questions, please feel free to use the chat, enter them, or ask your questions at the end. Dr. Joseph JaJa holds a joint appointment in the Department of Electrical and Computer Engineering and the University of Maryland's Institute for Advanced Computer Studies, UMIACS, at University of Maryland, and he's currently interim chair of the Department of Electrical and Computer Engineering. He served as the director of UMIACS from 1994 to 2004, as the interim vice president and chief information officer of University of Maryland's Office of Information Technology in 2010 to 2011, and as the co-technical director of the NIST FFRDC in cybersecurity from 2014 to 2015. Professor JaJa's current research interests involve big data, AI and machine learning, computational neuroscience, and high-performance computing. He is well known for his highly impactful 1992 book on parallel algorithms that's been cited thousands of times. I have my copy here. I like to carry it around with me. Great book. Still relevant after all of these years. He's published extensively in a number of areas, including parallel and distributed computing, combinatorial optimization, algebraic complexity, VLSI architectures, machine learning, and data science. He's received numerous awards, such as the Best Article in 2016 for Environmental Research Letters, the Best Paper Award for Scientific Visualization in 2012, the Internet to Driving Exemplary Applications or IDEA Award in 2006, and R&D Magazine's R&D 100 Award in 1997. In addition, Dr. JaJa is a fellow of the IEEE and a fellow of the ACM. During his 4 decades as a professor, Dr. JaJa has produced approximately 30 doctoral students, and he's got many academic grandchildren, great-grandchildren, even great-great-grandchildren, and I'm honored to be one of his doctoral students. His guidance to me, although it started 3 decades ago with my dissertation research, continues even today. One of the biggest things I learned from Joseph was to focus on solving real problems that make a difference to people, freedom, and society. Today, Dr. JaJa will be speaking on deep learning techniques to characterize dynamics in spatio-temporal neuroimaging data.
Well, thanks very much, David, for this nice introduction. And thank you for making me look so much younger. I don't know where you found this photo of mine. I'm delighted to be here and to talk about just a new work using neuroimaging data. This is an excursion for me into completely new domain. It's very different of flavor from any of the previous work that I have done. But this is what's nice about being in academia. You always shift gears and move into new areas as desired. But the other side of the coin is that you realize you're humbled by how little you know when you try to move into a new area. So I want to start by giving credit to my colleague, Professor Luiz Pessoa. He is the director of the Neuroimaging Center at University of Maryland and a professor of psychology, who introduced me to this area. Most of the work that I will be talking about, if not all really, is taken from the Ph.D. thesis of my student Manasij Venkatesh, who graduated a few months ago. So you know, What I will do, my presentation consists primarily of 3 parts. What I'll do is I'll stop in between the parts to see if there are any questions, and I can't see your faces. I don't have any way to tell whether you're following or you're sleeping. So I hope to be able to make at least a couple of breaks and to see if there are any questions. So the first part of my talk will be essentially high-level presentation about the overall area and the overall approach. It's a very high level. It's a little bit on the intuitive side, which means it's a little bit superficial. The second part focuses on one model that we have used for relatively small amounts of data, namely the reservoir computing model. And the third part will focus on much more complex data sets. And this makes use of the recurrent neural networks. So let me start with a kind of my first part. So the brain is a very complex system. I mean, one way to characterize it is by the number of neurons, which is over 100 billion neurons, each of which is connected to thousands of other neurons. It's a very complex system. But this has been studied over many, many years. I mean, of course, now with the neuroimaging, we have much more effective tool, but this is not a new topic. I mean, this has been studied over many, essentially over a century. And we have a very good understanding of several facets at a high level of the brain. So usually cells that are close to each other, typically, not always, are very well connected, almost always perform similar functions. We can decompose the brain, we can parcellate it into regions, and we can identify each region as being responsible for certain cognitive, perceptive tasks or motor tasks. So at a very high level, we have this very high, very coarse understanding of the brain, which has been developed over many years. More recently, as if 30 years and on, we have these very powerful non-invasive techniques called neuroimaging techniques that allows us essentially to look inside the brain and try to discover spatio-temporal patterns in the brain. And there are many different modalities. Here I listed some of these modalities: EEG, MEG, computer tomography, CT, and so on. However, this talk will focus on the magnetic resonance imaging. So essentially, it's a signal processing technique. So essentially what you do, you apply a signal, you measure the output, and then based on the output, you try to figure out what's going on in the black box in this case being your brain. So for MRI, it uses a very powerful magnetic field. It's non-invasive, and some radio waves. We can do lots of tricks to obtain many different types of MRI images. So in this talk, I will be focusing primarily on the functional MRI, which essentially creates what's called activation maps involved in your brain when the subject is doing certain tasks. So I'll go a little bit in more detail in the next slide. So, so this is a little bit a busy slide. If you look at the right-hand side, so we have the brain, and the measurements that we get from MRI for functional MRI, which is the right-hand side of this, essentially that gives us information about the overall cortex at the resolution of 1 to 2 millimeters. So you have essentially these voxels, these very small cubes. relatively small cubes. And we have information derived from MRI. Functional MRI, the information and the measurements that we have after some pre-processing, so I'm skipping on lots of steps here. After some pre-processing, we have activation levels. This is the amount of oxygenation, blood level, when a subject is doing some tasks. So this gives us essentially the time series of the activation levels. The time resolution is 1 second, 1 or more seconds. And essentially what we get for each of these voxels that are a part of the brain, so we essentially impose this 3-dimensional tessellation on the brain. You obtain voxels for each piece of the brain that is of spatial resolution, roughly 2 millimeter by 2 millimeter by 2 millimeter. So we get a time series of activation levels of each of these voxels. Usually we have around 100,000 to 140,000 voxels. And these time series of the voxels represent the activations of that part of the brain during the period when the subject is doing certain tasks. So what we do, of course, we have 100,000. We can't deal with 100,000. What we do, is decompose the brain into regions of interest. These are fairly large chunks. And then for each chunk, we average the time series, the activation time series. So we obtain usually 300 to 500 regions of interest, each of which now is characterized by this time series of activations. And then we can construct a network. This is very common. For example, to construct a network, each node represents one of these regions of interest. And then the edge connecting two nodes represent the correlation between the time series corresponding to the two endpoints. So this is called a functional brain network. We can also use different types of MRI data called diffusion tensor imaging, which essentially reflects the flow of water molecules in the brain, which means it captures the structural properties, the fiber connectivity, the white matter of the brain, and do something very similar and obtain a structural network. For this presentation, I'm going to focus on the functional network and try to understand how the brain reacts under certain conditions. And I'll be more explicit about what I mean. So a little bit more about fMRI data, since we will be using this extensively. As I mentioned, the brain is tessellated. You have this around 100,000, let's say, voxels, each of which now we have is characterized by a time series. The temporal resolution roughly is 1 to 2 seconds. And as I mentioned before, this is 100,000 voxels. Let me just make sure I mentioned that each voxel, even though you can think of it as very small, it's, what is it, 1 millimeter by 1 millimeter by 1 millimeter. But each voxel really contains around 100 million neurons. So this is not a very fine spatial resolution. This is like very coarse. But this is the best we can do. I mean, the technology is improving. We'll probably soon be able to do a sub-millimeter resolution. We already do it in some for specialized equipment. But the idea is to run experiments with subjects, with people doing certain tasks, and then collect this fMRI image about the various subjects. Of course, you have to be careful about the demographics so that it's you have a good representation of the population that you're trying to model. So what is it that I'm interested in, and how did Professor Luiz Pessoa get me interested in this? So right now, it's not easy to capture brain dynamics. So what is the brain dynamics? Well, at least the way it was explained to me is, suppose you're faced with a sudden danger, or you're under sudden emotional stress. Your brain somehow gets reconfigured to address this challenge that you're facing immediately. So we're trying to understand the dynamics of the brain, how it gets reconfigured so that it addresses a given challenge. The typical strategy used is what you do. I mean, you start with the usual strategy. You tessellate your brain into regions of interest. There are lots of very good techniques to do this. Usually we end up with 300 to 500 regions of interest. You average the activation time series for all the voxels within each brain regions of interest. So you get these time series. One way to try to understand brain dynamics would be to split this time series, which could be of length. I mean, resolution is 1 second. So you can decompose it into small time windows. And for each time window, you build the functional network. And then to try to determine the brain dynamics, you try to figure out how these functional networks, each of which is built over one small time window, how they changed, how they evolved. You can do some clustering techniques, you can define states. So this is a fairly commonly used technique. However, brain processes, at least in the cases we're interested in, are extremely dynamic. They change very quickly. It's not clear how you define this time window. And it's not clear, in fact, how you figure out the changes in the functional brain networks, the functional brain networks. So is there a way to capture the brain dynamics in a different way to get a better understanding of the underlying processes for the particular conditions or tasks that we're looking into? So that's at a very high level, the problem that we're trying to understand. So next, what I will do, and I'll stop in a couple of minutes. So next, what I will do, give you our overall approach at an extremely high level. So the brain is a very complex, very high dimensional, nonlinear dynamical system. So it's very hard to figure out what's going on given the current techniques. Let's suppose, just to motivate what I'm doing, we have only 3 regions of interest. I mean, of course, that doesn't make sense. But suppose instead of having hundreds or thousands of these regions of interest, suppose we only have 3 regions of interest. Then in this case, what we can do, we can really very easily visualize the dynamics by mapping these time series into trajectories. So when the individual is doing task A, for each time point, we draw the corresponding 3-dimensional points corresponding to the activations of the 3 regions. So for task A, for example, we can really, trace the state of the mind. In this case, it's ridiculous, it's 3 regions of interest through these trajectories. And when we compare task A to task B, we can clearly see in this case that the trajectories, the dynamics are very different. So this gives us, at least visually, a very effective way in trying to understand how the brain changes dynamics using visualization. Now, of course, so that's our overall goal is to model the brain using a highly complex nonlinear dynamical system, and then map the state into low-dimensional trajectories and try to use these trajectories as a way to understand the dynamics of the brain, use them essentially as signatures for any particular task as the temporal dynamics of the brain. So that's at a very high level, the overall approach. Now, of course, this is extremely high dimensional. So how do we model it so that we capture the characteristics of the tasks under study? And how do we do this dimensionality reduction so that we can look at the trajectories to try to understand the brain dynamics? So that's the overall approach. So next, I want to just motivate at least the first approach that we follow. So what if we want to classify, simple way to do classification is really to use linear classification. If we're lucky to have data that is more or less linearly separable, not quite, but more or less, then there are lots of very good, very effective techniques. I mentioned here a few of them: logistic regression, support vector machine, linear discriminant analysis, and there are many more. I mean, but these are probably some of the most widely used. But what if our data is not linearly separable? So one common technique is to use what's called the kernel trick. So you have a random projection into much higher dimensional space, and with a certain probability, your data will become more or less separable. So one of the strategies we're going to use is to use random projections. And then, which means we're dealing now even with a much higher dimensional dynamical system. But it's much easier to analyze the system and then map it do the reverse operation with dimensionality reduction, and to essentially try to figure out these trajectories. So I think what I'm going to do, I'm going to stop here. So this is kind of a very high level, essentially high level overview of kind of the overall plan and the problem that I'm trying to address and the type of data that I'm using. So since too bad I can't see you or see your faces, I'm going to stop here and see if you have any questions about this first part of my presentation.
Maybe I can just ask one question and others may chime in or use the chat.
Sure.
For the data rates of the fMRI, what are they typically like, and what is both the volume and the rate of the data that you typically see?
So as I mentioned, the spatial resolution is roughly 1 to 2 millimeter cubed, which means you have around, so for each individual, so you collect data about subjects, and usually would love to have thousands. And we're starting to have enough information now for each task with thousands of people. So for each individual that does a certain task, you have a volume consisting of roughly 100,000 to 150,000 voxels. For each voxel, we have a time series of length 100 to 150. The time series resolution is typically 1 second temporal resolution. So for each individual, we have 3D volume with 150,000 voxels. Each voxel is characterized by this time series. Does that answer your question, David? It's a lot.
That's good.
It's a lot of data for one individual in one task.
Mhm. Thank you.
Yeah, sure. And it's very high dimensional. So let me move to the next part of my presentation.
Joseph, there's a question in the chat. Can you repeat why we need to know in advance activation regions? And a second part, given that we have the dynamics of brain projections you mentioned, can you give an example of potential applications?
Oh, well, first, the first question, what was the first question now? We don't know the brain regions. What we do, we have a very coarse idea about different brain regions at a very coarse resolution. For example, we know, for example, the parts that's responsible for our vision system. I mean, this is very big area. We're interested in much finer resolution than this resolution. This is not even 1 millimeter. This is much, much larger. The application, the initial application I start, well, first of all, trying to understand how the brain works in itself. It's a very fascinating area, and you can imagine the application. But what got me initially started, it's not this work, is trying to figure out brain disorders. What happens in the brain when you have certain brain disorders, for example, or cognitive problems? What exactly, which parts of the brains are impacted? And of course, ideally, what we would like to do is figure out how to solve this problem or find a way to ameliorate these conditions. So the main applications is trying to better understand how the brain works so that we can have a much better way of handling, let's say, dementia, different type of mental disorders, cognitive disorders, and so on. So that's the initial motivation. But just trying to understand the brain and how it works, I mean, this is such a fascinating field in itself. That, you know, it's definitely worth studying extensively.
Thank you. I think we can move on.
Okay, so so this is the second part of my talk. So before I introduce the model that I am using, and I promised you these, you know, complex dynamical systems and trajectories. Before I get there, you know, most of the work is empirical. I mean, you know, this is again very different than any of the previous work that I have done. So I need to define the data that I'm using to essentially not only motivate, but also to justify the work that I'm doing and validate it. So the first task that I'm looking at is, and these are from the Human Connectome Project data. This is available online. This has been going on, I don't know, maybe for 15, 20 years. It has an extensive data sets of neuroimaging data, extremely well annotated, well calibrated. They do really very nice pre-processing so that we don't have to deal with any of these issues. So the first data set that we'll look at is called the working memory. So the working memory, I want to start with two tasks related to working memory. The first task is called zero back working memory task. So this task, what you do up front, you tell the individual or the subject, look at this image, let's say image of a car. And then you show them a sequence of images, and they should tell you whenever they see that image. So you tell them up front, this is an image of a car. Here it's denoted X. Whenever you see this image, and I'm going to show you a sequence of images as I'm taking the fMRI data. whenever you see this particular image, you can hit a button or raise your hand or whatever. So this is called zero back. The second task, memory task, is called two back. You don't have a fixed target. What you ask each individual is to hit a button whenever the image they're currently looking at is the same as the one they saw 2 steps before. So in this case, if you look at this L, in this case, you raise your hand or click a button, or if you have a joystick, you push it. You see L, you had seen it 2 steps before. So this is called two back work in memory task. So we have 2 tasks here, and we have data for, and now I want to show a different type of data. This is called theory of mind. These are kind of video clips of geometric objects, typically rectangles or triangles, that they are moving. Some of the interactions could be completely random. So let's look at this. I mean, you can't figure out what's going on here. I mean, that seems fairly random. But if you look at this video clip at the bottom, let's see if you can figure out whether there is some social interaction, there is some meaning that you can assign to this. You may have noticed that the red triangle was trying to figure out how to get off its jail or box, and the blue triangle helped him find the door. So clearly there is much more meaningful information, much more social interaction. On the other hand, you had to figure this out. You couldn't figure it out immediately. Maybe if I can show it again, you couldn't figure this out. So your mind has to work. Dynamics now come into play, so that you're trying to figure out whether there is indeed some meaning, some social interactions. So in this case, for the work in memory, it's a relatively stable mental state for both zero back and two back. For this, your mind had to work continuously until you figure out whether it's random interactions or social interactions. So these are the 2 tasks we'll be looking at. I hope I convinced you that for the work in memory 2 tasks, essentially there's relatively little dynamics involved. Where for the theory of mind, social cognition task, that it takes some effort. The brain has to work to figure out whether or not there is indeed some meaningful social interactions. It takes some time to figure this out. So how are we going to model these using Fmri data from these 2 tasks? So I'm going to use a very specialized model called Reservoir Computing, which is a very special type of recurrent neural networks. So let me first define it at a high level. So this will be my high-dimensional dynamical system, and I'll tell you later how I'm going to do dimensionality reduction and how I'm going to derive these trajectories as I promised. I keep my promise. So, but let me first define this reservoir computing model. So essentially, the reservoir computing model consists of 3 parts. There is an input layer. This is similar to a recurrent neural network called reservoir with feedback loop. So unlike deep learning networks, it's essentially feed forward. Here you have these feedback cycles, which means the state will be defined by the values of these neurons in the reservoir. So the next state, not only does it depend on the current input, but it depends on the previous state. That's why you have feedback loops. This is called reservoir. And we have a readout layer. So reservoir computing consists of these 3 parts. So the time series, these are the activations. This is derived from the Fmri data after we pre-process the data and we create these ROIs, the regions of interest. We have, let's say, 300 of these time series. At each 1 second, we have 300 essentially value activation levels. So we feed this into our reservoir. And this reservoir, essentially, the way I'd like to look at it is it's creating a random high-dimensional projection. But this random projection depends on the past. This is not like a deep neural network where for each layer it depends on the input provided from the previous layer. Here you have this feedback loop. And the last part, what's amazing about this model is that the weights of this reservoir of this specialized recurrent neural networks are not trained. And they are, I'll tell you a little bit more how they are determined in a minute. They are fixed, determined before we start running our model. The same thing, in fact, applies to the input layer. These are randomly selected in a special way, and I'll say a little bit more in the next slide. The only layer that's trained is this readout layer. This is the only layer, the only weights that are trained using the data and the particular task we're looking at. So the classification in this case, for example, for the memory task, would like to know whether this is 0 back or 2 back. So this is a classification problem. So what we would like to do is train a reservoir to essentially distinguish between these two tasks across all the individuals in the data set. So the only layer that's really learned are the weights in the readout layer. Everything else is fixed. So this is computationally extremely efficient. Once we figure out which weights to assign to the reservoir and the input layer, it's extremely efficient and works very well even with small amounts of data. So let me say a little bit more. So we represent the values of all the neurons as a vector representing the state of the reservoir denoted by x of t. U of t represents the input. These are the activations of the 300 regions of interest at each time t. So, and then there is a readout layer here, which does classification for our purposes. So the next state is defined by these equations. So the intermediate state, x hat, I mean, this is nonlinear. So 10H, we take the input, we apply this, the input weights defined by the matrix Wn to the inputs. And we apply this reservoir matrix W to the current state so that we obtain the next intermediate state. So more precisely, this W is within reservoir matrix that is predefined, it's fixed. And let me say a little bit more, it's random, it's sparse, but it has to satisfy some properties. So the spectrum of this matrix has to be less than 1, i.e. if you look at the absolute value of all the eigenvalues, they have to be less than 1. This is important. It's called the echo state, because what you'd like to do is you'd like to take into consideration the previous state, but you don't like the previous long history to really dominate. So you need this condition. And then we're mapping this randomly into a higher dimensional dynamical system. So the number of reservoir units will be T or tau times number of input units. So we have this parameter tau that we have to determine. Wn is the input reservoir matrix, which is determined randomly, but it's a dense matrix. So W and Wn are predetermined. There are all sorts of constraints that you have to satisfy when you determine these. And the system works by essentially by using this equation to determine the next state. So next state, there is this forgetting rate, another parameter that you have to select carefully. So it depends on the previous state as well as the intermediate state. You want to make sure the intermediate state doesn't dominate, and then you have very little, the previous state has very little impact. So we have a couple of parameters to deal with, namely T, which determine the size of this reservoir, the dimensionality of this reservoir, and alpha, which determines the forgetting rate. How much should you take into consideration the previous state? So that's a little bit formal introduction of the reservoir. So what I'll do next is start by applying this model. Here I'm doing just pure classification. I'll get to the dynamics in a minute. So, but I want to make sure first that our model does capture the essence of the problems or the tasks that we're looking at. So the first question is, if we use this reservoir computing model, how well can we classify 2-back versus 0-back in working memory? How well can we classify social versus random in theory of mind data set? So before I go on, this readout layer, I'm using logistic regression for classification. And this is the only layer that's straight. These are the only weights that are learned. So we have 200 subjects for each, the working memory and the TOM data set. And what we will do next is I want to show that this reservoir, the state, this dynamical system defined by this reservoir computing is better at doing classification for both tasks than using the pure activation levels. So let me now show some results. So first for the working memory, so I modify the parameters. I showed the parameters tau, which reflects the dimensionality of the reservoir, as well as this alpha, the forgetting rate. So different colors correspond to different values of alpha. So for the working memory, the green light represents, if we use activation and logistic regression, this is the what we get, the accuracy we get for classification is around, I don't know, 79% accuracy. On the other hand, if we use the reservoir state, plug it into the logistic regression network, we get much better accuracy. As you can see for the working memory, really the size, if we modify the size of the reservoir, the accuracy doesn't improve. So the size, this is tau reflected by tau here. Tau is equal 1, same size as the ROIs. 2 is twice as large, 5 times as large, 10 is 10 times. So we really don't get much improvement. On the other hand, for the theory of mind, first we get much better accuracy using the reservoir, but also we get much better accuracy as we increase the reservoir size. So if it's 10 times as large, so we start from 300 regions of interest, and the dimensionality of the state of the reservoir state is 3,000, we get much better accuracy in terms of classification. Now, since we're feeding the data sequentially, one second at a time, we can look at the accuracy as a function of the time. So when we look at the working memory, initially, of course, the accuracy wasn't so good. I mean, it was reasonable. But after a while, it remained flat, more or less. On the other hand, if we look at the working at the TOM data set, the theory of mind, initially the accuracy was at chance level, but suddenly, as the brain figures out what's going on, try to figure out whether this is random interactions or the social interactions, there is some meaning to it, the accuracy improves substantially, and it gets, in fact, over 90% accuracy. So at least this gives you an indication that the reservoir model is a good way to model at least to make the difference between the two tasks, the working memory and the theory of mind. And the theory of mind, it seems to capture the dynamics in a very significant way. But as I promised, I mean, the goal really is to map this into lower dimensional trajectories and try to visualize what's going on. So how do we take the reservoir state and map it into much lower dimensional space and still maintain more or less the same classification accuracy and map it into trajectories? So this is what I'll go over next. So first, what we take the reservoir state and we do a linear transformation using PCA. PCA, principal component analysis, is usually used to reduce the dimensionality. Here, I'm not reducing dimensionality. All I'm doing is removing correlation and try to remap this time series into a new orthogonal basis. So I have exactly the same dimensionality as before. And then what I will do, I'm applying now the logistic regression. But what I do, I take only the top K values for some value of K. The largest K weights of the logistic regressor, or the bottom K. The top K really are biased toward one decision. Let's say 2 back, and bottom K are biased toward 0 back. So I take this highly dimensional dynamical system, map it into just K values, either top or bottom K. And now what I want to show is how well can I do with just using these K values. So if we look at the reservoir data and we apply this technique for dimensionality reduction. So if we use the reservoirs dimensionality for working memory, we achieve, I mean, I showed this in the previous slide, we achieved 87% accuracy. For the theory of mind, we achieved 92% accuracy. Now, if I use just K is equal to 2, I achieve 78%, which is better, by the way, than just using the activation values. As we increase the number of dimensions, the accuracy improves. Really, even for dimension 12 or 14, we achieve almost the same accuracy as using the full dimension of the reservoir data, the 3,000 dimensional data produced by the reservoir. And similarly, for the theory of mind, we achieve very good performance even for K is equal to 2. For K equal to 12, we achieve, very good. I mean, it's 87% accuracy for theory of mind to distinguish between random interactions and social interactions. Had we used the activation data, first, the accuracy would have been much smaller anyway. It would be 78%. As here we have 87%, much less. And we need many more dimensions using exactly the same technique to achieve it, to come even close to this. But anyway, what this illustrates is that this dimensionality reduction technique, starting by remapping the data, the reservoir state, using PCA, and then using these k largest weights of the logistic regression, is an effective technique for reducing dimensionality and without sacrificing anything significant for small values of k in terms of accuracy, classification accuracy for the two tasks. So next, what I will do, as I promised, I want to go back to trajectories. And this is what I had promised. So let's suppose now we take k equals 3, as we have shown in the previous slides. K equals 3 or 4 or even 2 gets you very good accuracy. So it still captures the essence of the data. It still has much better accuracy in terms of classification than using the activations. The activations, the 300 activations produced by the Fmri Data. So let's map the data produced, of course, across all individuals. Let's map them into trajectories and three-dimensional trajectories using this dimensionality reduction technique after they had passed through the reservoir. For working memory, so we're trying to distinguish, our model captures the differences between 0 back and 2 back, its classifier. So if we look at the corresponding trajectories for working memory, you will see that the trajectories from the very beginning were different. Somehow your mind was in different states for 0 back and 2 back. However, if you look at the theory of mind, so we're trying to distinguish between social and random interactions. Initially, the two trajectories are almost identical. It's not until after 10 seconds or so that you start to distinguish it. You start to say, oh yeah, looks like more like a social interaction. So of course, this is a very low level. I mean, this is 3-dimensional. We started from reservoir state, you had 3,000 dimension and you mapped it in just 3 dimensions. But this in itself just show you the distinguishes between the two types of interactions, indicates that the dynamics are very different for these two tasks, for work and memory tasks and theory of mind tasks. Well, how is that related to neuroscience? So why don't we try to correlate the regions of interest to the decisions made by the logistic regressor? So we can have some correlation. These are the weights of the logistic regression. And this is the correlation between the regions of interest M and the corresponding weights from 1 to K. So in this case, if we do this for working memory, we figured out the regions. These are the regions of interest that are responsible for distinguishing more or less between 0 back and 2 back. We use this for the working memory. So it turns out that, I mean, this is where I have to depend on my Luiz, my colleague. So if you look at the regions identified as really strongly correlated to deciding whether this is 0 back or 2 back, you will find this is lateral prefrontal cortex, parietal cortex, interior insula, and these are the regions that you would expect. For Tom, you identify different regions that led to this decision. So, of course, whatever we do in terms of modeling, we want to project it back into neuroscience, try to understand what's going on the brain. There is much more work that we did around this area, but this is not my area of expertise. I want to focus on the machine learning techniques. So that concludes the second part of my talk. So I want to stop here and see if there are any questions about the second part. So I see 3 questions in the chat box.
So there's one question or comment in the chat from Jaden Kashyap, who says, thanks for choosing such an exciting topic for this talk, Dr. JaJa. In addition to yourself, what may be other groups in the world that a beginner in the field can follow who are doing cutting edge research and studying brain functions through computation, non-invasive approaches? Thanks for the talk.
Certainly. I mean, this is a big area that has been studied extensively over the past 15 to 20 years. Well, much longer, but I'm looking at the relatively high resolution. While high is like 1 to 2 millimeter, that's not high. Temporary resolution is okay, 1 second. You'd like the order of milliseconds. Sometimes some of the mental processes are much finer resolution. So there is a really very extensive literature in this area. For Fmri data, the signal is, this has also been studied extensively. So this is usually leads to functional networks, as opposed to structural networks. Lots of graph theory has been used to model these networks, understand the structure of these networks. I mean, you have usually you remove the edges that have low values, low correlation. What you're trying to do is the weight on each edge represent the correlation between the activation time series of the endpoints. If it's small, you can remove this edge. So you end up usually with relatively sparse networks, and you try to understand the various structural properties and topological properties of this network. This has been studied extensively. So there are many references. If you Google computational neuroscience, you'll find lots of references. Now, in spite of what I said, there is so much work to be done. However, I should mention that just as a caveat, this is not pure data science problem. You really need a collaborator from neuroscience so that your work is relevant to neuroscience. At the end, what you're trying to do is trying to have a better understanding of how the brain functions, how different regions interact with each other, what happens when you have a brain disorder, dementia, and so you need expertise, neuroscience expertise, so that the problem you're looking at is relevant to neuroscientists. They get excited. So whatever you try to do, you really need a collaborator from that area who really has a good grounding in essentially neuroscience to guide the research that you're doing. There is much more work that we did in this area, but this was led by Luiz Pessoa, and that I don't fully understand. I mean, this is not my area of expertise. But whatever, you can have a very fancy model, you can obtain beautiful empirical results, but these are meaningless unless they are well justified in terms of the neuroscience. The brain dynamics is a very important and challenging problem. And if you think about it, when we learn something new or when we make a decision, it's not just based on the current data that we have. There is a history that's embedded in our mind, in our brain, that we make use of. That's why we don't forget our native language. So trying to understand the brain dynamics and how it's evolving is very important, for example, for language acquisition. So, but there's a lot of work that has been done at different levels. It's a very exciting area, fascinating area. what we know is so tiny compared to what we should know about this area. And it will have an impact, both biomedical, societal. I mean, you know, cognitive disorders, mental disorders, you know, I mentioned dementia, but how about depression? And especially under this pandemic, trying to understand this and help people really. I mean, what else would you want in life if you can do something like this? Any other questions? David, how much time do I have?
So we're just over the hour, but we can continue if you have a few more slides. And
well, I have more than a few slides. So I think maybe I'll send you my presentation, because there is a part that deals with much more complex tasks than these two relatively artificial tasks. Maybe I should do, will it be okay if I do one more slide?
Sure, that's fine.
Okay, let me move to. So the data that I present, the two tasks, are relatively simple tasks. I mean, you know, these video clips that I showed about moving triangles, I mean, you know, these are fine, but they are artificial. If you want to study, you know, brain dynamics, one possibility is to have the subjects watch real movie clips. And these clips should really have, you know, be social, you know, kind of emotionally exciting, affective, can impact your brain. So there is such a data that was developed by the Human Connectome Project. So we used 15 short video clips, and each participant, and we had 170 in this data set, participants, watched each clip once. So the clips are very short, few minutes. So I'm sure you will recognize these movies. And I even watched some of them. So Home Alone, Inception, Star Wars, very short clips. So you watch these movies. So you have 15 now video clips. Each individual watch these video clips. Of course, you have to reset after each video clip because you don't want the Fmri data collected from the next video clip to be impacted by the previous video clip. So anyway, we have all of these data from 176 participants, each of which has watched 15 video clips. And we needed to model this to try to capture brain dynamics, create these trajectories. We had to use a much more sophisticated model than the reservoir computing model. And we used a combination of this is where the deep learning comes into play, recurrent neural network, full-blown recurrent neural networks, and a very different type of dimensionality reduction. And we were able to do something very similar to what we did for the memory test and for the social interaction theory of mind test. So I think I'll stop here. I'll be happy to answer any questions you may have. And thank you again for inviting me to the seminar. So I guess I'll.
Thank you again for a wonderful seminar. It's great to see data science being used for something as important as understanding brain function. Let me see, are there any
brain disorders?
Are you able to work on the inverse problem? So given a brain injury, try to understand what impact that has on the brain function?
No, but something related, which of course I didn't have the time to present. With these video data, each individual was essentially asked to answer many questions. We have a lot of information about, let's say, verbal IQ tests, various cognitive tests, and so on. And the model that we use can predict these values based on the fMRI data from video clips. For certain video clips, turn out to be much more effective in predicting your personality, so to speak, like, which is amazing. We don't fully understand this, but the problem you mentioned is very interesting, and it's lots of practical importance. But it's hard to get data for it. You see, the nice thing about fMRI is non-invasive and has no side effect. So you can tell people to sit down, watch a movie when you're collecting that fMRI data. But with brain injuries, keep me away from this. I don't want anybody to be hurt. There is some invasive techniques that are used on mice. And, it's really kind of a little bit cruel. But the resolution, spatial resolution, is like at a neuron level. Here we're talking about millions of a neuron for each box.
So there's a question from Grace G. Are the data sets on brain activity available for public research, say multiple channel data sets?
Absolutely. These are all, go to the Human Connectome Project website. And these have been extremely well curated. I mean, it's easy to make mistake pre-processing the data before you get these activations and do the tessellation. You don't want to do this. You want the professionals to do it. There are extremely well-defined, well-understood pipelines that have been used to generate this data, and they have been validated by many researchers. So go to the Human Connectome Project website. You'll find lots of data sets. Very few, though, involves real dynamics, because when you want to try to do is have somebody in stable mind to figure out, what functions of the brain are responsible for certain tasks. So you don't want the brain to move from one state and jump to any other state. That's why brain dynamics is kind of a tough topic to deal with.
So I know that these are large data sets that are produced. Do you use any high performance computing, such as FPGAs or GPUs?
Yeah, we use GPUs. Yeah, I mean, otherwise we can't do the work. For even for the reservoir computing, but reservoir computing is not a big challenge. But for the third part, for the video clips, we use recurrent neural networks and LSTMs, plus dimensionality reduction, plus fully connected network. These you needed to use GPUs. Yeah, absolutely. Without GPUs, you can't survive.
Right. Well, thank you again for this.
Sure, my pleasure. Yeah.
And thanks to the audience, both here on Zoom and on YouTube, and for sharing this seminar series this academic year, as this is our final seminar for this semester. And so thank you again, Joseph.
Sir, my pleasure. My pleasure. And thank you for inviting me.